Welcome to Biology at Priyajit, my dear viewers. Today we explore one of the most important metabolic pathways, beta-oxidation of fatty acids. Just as a power plant breaks down fuel to generate energy, our cells also degrade the fatty acids step by step in mitochondria to release acetyl-CoA, NADH and FADH₂ for ATP production. So this process forms the backbone of lipid catabolism and energy metabolism, especially during fasting and exercise. So let us understand its steps and significance in a simple and systematic way. So, introducing fatty acid oxidation. Oxidation of long-chain fatty acids to acetyl-CoA is a major source of energy, especially in heart and liver, up to 80% of energy. So, electrons released during this process pass through the respiratory chain to produce ATP. So, and acetyl-CoA enters the citric acid cycle for complete oxidation to carbon dioxide and H2O. In liver, acetyl-CoA can also form ketone bodies which supply energy to the brain during glucose shortage. In plants, acetyl-CoA is mainly used for biosynthesis rather than energy. So why are fats good energy stores? Triacyl glycerols or fats are highly reduced and yield about twice as much as energy as carbohydrates. They are insoluble and stored without water, so they do not increase cell osmolarity. They are chemically inert, allowing safe long-term storage. So how are fatty acids are oxidized? Before proper oxidation, fatty acids are activated by coenzyme A. Then they are broken down in mitochondria by repeating four-step processes called beta-oxidation, which removes two carbon units as acetyl-CoA, and this complete oxidation occurs in three stages. First one is the beta-oxidation, which produces acetyl-CoA, then it goes to citric acid cycle to produce carbon dioxide, and the respiratory chain then ultimately produce the ATP. Activation and transport of fatty acids into mitochondria. Enzymes for fatty acid oxidation are located in mitochondrial matrix. The short chain fatty acids which are less than or equal to 12 carbons enter mitochondria directly. Long chain fatty acids which are more than 14 carbons, they need the carnitine shuttle to enter. Activation of fatty acid. Fatty acids are activated on the outer mitochondrial membrane by acyl-CoA synthetase. Reaction fatty acid plus CoA plus ATP will produce fatty acyl-CoA plus AMP plus 2PI. So this forms a high energy fatty acyl-CoA using energy from ATP. Formation of acyl carnitine. The acyl group is transferred from CoA to carnitine by carnitine acyl transferase 1 or CAT1. This forms fatty acyl carnitine which can cross the inner mitochondrial membrane. Entry into matrix and regeneration of acyl-CoA. Acyl carnitine is then transported into the matrix by a specific transporter which is known as carnitine shuttle. Inside carnitine shuttle there is carnitine acyl transferase 2 or CAT2 enzyme which transfers the acyl group back to coenzyme A, forming fatty acyl-CoA again. Then free carnitine returns to the cytosol. Importance. This step system is called the carnitine shuttle. It is the rate limiting and regulatory step of beta oxidation. Once inside the matrix as fatty acyl-CoA, the fatty acid is ready for beta oxidation and ATP production. So this is the image showing the migration of fatty acyl-CoA with the help of carnitine acyl transferase located on the outer mitochondrial membrane. Then it will cross the intermembrane space, and in the intermembrane space there is a carnitine channel. It will transport the channel with the help of carnitine acyl transferase 2. It will again transformed into acetyl-CoA. So fatty acid entry into mitochondria. This is the image showing the fatty acid entry into mitochondria and their regulation. Malonyl-CoA inhibits carnitine acyl transferase 1. So these prevent the fatty acid breakdown when fatty acid synthesis is going at the same time. Mitochondrial oxidation of fatty acids. There are three main stages. Stage 1, beta oxidation, where fatty acid is converted into acetyl-CoA. Fatty acids are broken down two carbons at a time from the carboxyl end. Example, palmitic acid undergoes 7 cycles of beta oxidation. Each cycle removes 2 carbon or acetyl-CoA. After 7 cycles, all 16 carbons form 8 acetyl-CoA molecules. Each acetyl-CoA formation then removes 4 H atoms, that is 2 pairs of electron via dehydrogenases enzymes. Second stage is the citric acid cycle. From acetyl-CoA, carbon dioxide and electron carriers are formed. Acetyl-CoA then enters the citric acid cycle in mitochondria, and same as acetyl-CoA from glucose, it produces NADH and FADH₂, which carry electrons to the next stage. Electron transport chain, electron carriers when it will produce ATP, then NADH and FADH₂ donate electrons to the mitochondrial respiratory chain, and then electrons reduce oxygen to form the H2, and energy is released, is captured as ATP. Beta oxidation of saturated fatty acids. The breakdown of fatty acids in mitochondria happens in 4 main steps, producing acetyl-CoA for energy. Here is how it works. First one is the dehydrogenation. The fatty acyl-CoA is first attached to coenzyme A, forming fatty acyl-CoA. An enzyme called acyl-CoA dehydrogenase removes electrons, creating a double bond between C2 and C3, forming trans delta 2 NOL-CoA. There are 3 types of this enzyme depending on chain length. There are very long CAD or acyl-CoA dehydrogenase, MCAD, medium chain acyl-CoA dehydrogenase, and small chain acyl-CoA dehydrogenase. So electrons go to FAD, then go to the electron transport chain and producing about 1.5 ATP. Second stage is hydration. Water is added across the double bond by NOL-CoA hydratase. This forms the L beta hydroxyacyl-CoA or 3-hydroxyacyl-CoA. Think of it is like filling the double bond with water. So this is the 4 stages of beta oxidation. First one is the dehydrogenation, second one is the hydration, third one is the all again dehydration, and fourth one is the thyrolysis. So third stage, this is also dehydrogenation. The hydroxyl group or OH is oxidized to a keto group by beta hydroxyacyl-CoA dehydrogenase, forming beta keto acyl-CoA. Then NAD plus takes the electron and producing NADH to ATP. And then the last stage, there is thyrolysis, where splitting occurs. An enzyme called thiolase splits off 2-carbon acetyl-CoA from the end of the fatty acid. The remaining fatty acid is then shortened by 2 carbons and goes back into the cycle. So for long fatty acid chains where more than 12 carbon, these steps are done by a trifunctional protein TFP complex, which keeps enzymes together for efficiency. And each round of beta oxidation shortens the fatty acid by 2 carbons, producing acetyl-CoA, NADH and FADH2, and all they are energy carriers. So dehydrogenation, hydration, dehydrogenation and thyrolysis. So remember DHDT for the sequence, which is helpful for the students for their remembrance. Beta oxidation, this is the repeating the 4 steps of to produce energy when a fatty acid like palmitate or 16 carbons is broken down. Each round of beta oxidation removes 2 carbons as acetyl-CoA, and palmityl-CoA 16 carbon goes through 7 rounds of beta oxidation, producing 8 acetyl-CoA, electrons in FADH2 and NADH, and some water. Stepwise energy yield, that is 1 round. It will produce 1 molecule acetyl-CoA, FADH2 1 molecule, which will generate 1.5 mole ATP, and NADH 1 molecule will generate 2.5 mole ATP. So total at ATP per 2 carbon removal is 4 ATP. So in case of palmitate 16 carbon, there are 7 rounds of beta oxidation produces 8 acetyl-CoA, so there would be 80 ATP, and 7 FADH2 is 10.5 ATP, and 7 NADH is equivalent to 17.5 ATP. So total ATP is 108 ATP will be produced from palmitate. Acetyl-CoA then enters the citric acid cycle and energy yield from 8 acetyl-CoA. It will output is 8 acetyl-CoA, 80 ATP, 16 carbon and 16 molecules of H2O. This is the overall energy from complete oxidation of palmitate. So there 108 is net gain, and 2 ATP will be utilized to convert palmitate to palmitoyl-CoA. So net gain 106 ATP per palmitate molecule. Oxidation of unsaturated fatty acid. Most fatty acids are unsaturated, which contain cis double bonds. So the normal beta oxidation enzyme, that is NOL-CoA hydratase, works only on trans double bonds. Therefore, 2 extra enzymes are needed: NOL-CoA isomerase, which converts cis double bonds to trans, and then 2,4-dionyl-CoA reductase helps in polyunsaturated fatty acids. So oleic acid, it will finally 9 acetyl-CoA molecules. And the linoleic acid, it will finally produce 9 acetyl-CoA molecules. So this is the process. So summary: unsaturated fatty acid need extra enzymes to rearrange or reduce the double bonds so that normal beta oxidation can continue. This is the reaction cycle of oxidation of monounsaturated fatty acid, and this is the pathway displaying the oxidation of polyunsaturated fatty acids. So oxidation of odd chain fatty acid, most fatty acids have an even number of carbons, but some in plants like marine organisms and ruminants there are odd number of carbon. Beta oxidation of odd chain fatty acids proceeds normally until the last step. So the final products are acetyl-CoA and propanoyl-CoA. So the conversion of propanoyl-CoA, it needs three extra steps: carboxylation, but propanoyl-CoA will be converted into D-methyl-malonyl-CoA, then epimerization, the D-methyl-malonyl-CoA will be converted into L-methyl-malonyl-CoA, and then rearrangement, L-methyl-malonyl-CoA will be converted into succinyl-CoA. So this is the reaction cycle. There are three stages. The final step, succinyl-CoA then enters the citric acid cycle for complete oxidation. So regulation of fatty acid oxidation in the liver, it enter mitochondria for beta oxidation for energy production and be used in the cytosol to make triacyl glycerol and phospholipids for storage. This is the key control point, that is carnitine cycle, role of malonyl-CoA, and regulation by the energy status. This is the genetic defects in fatty acyl-CoA dehydrogenase enzyme. So my dear students, you have seen how beta oxidation of fatty acid is a key energy link pathways in cells. Though the stepwise breakdown of fatty acids into acetyl-CoA, it supplies fuel for the citric acid cycle and ATP production. For university and competitive exams like CSIR-NET, remember that understanding this process is essential for grasping the metabolic energy flow. So keep revising and exploring the wonders of metabolism. Thank you.